My name is Yu Tianxin. I will introduce the paper submitted to EMETA. The title is Plant Pathogenesis Towards Multidimensional Understanding of the Microbiome. The resident microbiota is a hidden game changer in plant health and disease. The invasive pathogen can hijack certain members of the resident microbiota to form partnerships within the reassembled community, which is currently conceptualized as the pathobiome. The pathobiome facilitates the pathological process of host plants from the onset of the disease to its progression by coordinating complex interactions at multiple scales. Additionally, an increasing trend of plant disease outbreaks has been observed, with more drastic trends of global change. The native plant microbiota mediates an interaction network at multiple scales. Such complex interactions determine the status of the host plants, with an implication on agricultural production and global food supply. The disease-preventing members from the resident microbiota, recently coined as soterabionts, are known to function as a defensive layer against pathogen invasion. However, some native members in the resident microbiota can also be potential pathogens. They even can be manipulated by the invasive pathogen to form the partnership along with the pathogenesis process. The host pathogenesis-associated microbial consortia is contemporarily conceptualized as pathobiome. A growing awareness of pathogenesis has evolved from the one pathogen, one disease paradigm to the pathobiome concept. Many diseases previously attributed to one pathogenic agent are likely to be the result of interactions between multiple microbial taxa and their host plants. Therefore, the mechanistic insight into the plant pathobiome-associated interactions is important for uncovering the process that governs the outcome of pathogen infection in the host plants. The mutualistic relationships among the invasive pathogen and the native potentially pathogenic microbial members are generally concealed within the complex intramicrobiome interaction, from intraspecies to interspecies and interkingdom, which negatively affect the homeostasis of vegetative and reproductive organs. Drive the infection outcome and result in disease progression. Quorum sensing is the first identified bacterial intraspecies signaling system, which enables bacterial cells to chemically sense the density of the surrounding population. Indeed, intraspecies communication in situ is largely impacted under a more sophisticated network, in which the diverse bacterial taxa resident in the same eco-niche of the host plants mediate interspecies interactions. Quorum quenching has been previously characterized as ubiquitous interspecies interaction employed by symbiotic bacteria to interfere with the QS of phytopathogenic bacteria. Establishment of the mutualistic relationships among the invasive pathogen and resident microbiota members highlight the ecological scale implication of the intraspecies communication on the plant pathogenesis. Therefore, further efforts are expected to explore the group behavior involved in pathobiome-coordinated infection in host plants. In plants, intramicrobiome interaction is not limited to the intra- and interspecies level, but is also featured by interkingdom interactions and bacterial-fungal interactions majorly, including physical interactions and chemical interactions. Investigation of the specific cooperation mechanism of the assembled pathobiome has further shown that the bacterium can physically attach to fungus and utilize fungus as an effective vector to facilitate its infection and expansion in the host plant. Moreover, the pathogenic microbiome can benefit from bacteriome through the formation of the multi-kingdom biofilm matrix, which functions as a physical barrier to protect against the host and antimicrobial insults and advancing the infection. Overall, the characterization of the microbial-derived signaling molecules, ranging from chemical small molecules to larger effectors, would be instrumental in revealing the landscape of BFIS-mediated plant disease progression. In addition to intramicrobiome interaction, the pathobiome-mediated interaction is omnipresent at multiple scales. The potential pathobiome members collaborate to counteract the plant genetics-governed alliance. For example, effectors produced by different members of the plant pathobiome can rapidly evolve to overcome ETI by evading recognition of our proteins. leading to effector-triggered susceptibility, and these effectors may target various plant factors encoded by susceptibility genes to magnify the ETS of the host plants. Furthermore, the pathobiome members may arm with diverse small molecule-type virulence factors to subvert plant innate immunity-based defense strategies. Interestingly, a tritrophic interaction study demonstrated that the specific members of the plant pathobiome possibly promote their spread and disease epidemics via manipulating the host attraction to insect herbivore vectors. Additionally, the plant pathobiome members may compromise defense against insect herbivores by influencing the crosstalk between the phytohormones jasmonic acid and salicylic. These findings could explain the limitations of the conventionally one pathogen-targeted approach. Hence, the characterization of the plant pathobiome members at multi-scale will serve as an important basis for the novel strategies to break both pathobiome-responsible pathogenesis and insect pest damage. A critical question remains unanswered as to what and how the certain circumstance triggers the plant pathobiome prevalence. Global change has been predicted to continue with more drastic trends since the last century, and the plant disease epidemics arise under this scenario. It is time to pay attention to implication of global change on the prevalence of the plant pathobiome. Climate change and anthropogenic activity may influence the composition and microbial community diversity of phyllosphere microbiome in the water and soil. Prevalence of antibiotics and microplastics could increase the abundance of potential pathogens. The increased inputs of agrochemicals and fertilizers cause uncertain impacts on the structure and function of the resident microbiota in both rhizosphere and phyllosphere, and reversely allow the non-native potentially pathogenic microbes from the surrounding environment to colonize the below to above niches of host plants through a vacuum effect. These findings indicate that the natural and human triggers are likely to play important roles on the plant pathobiome prevalence under the scenario of global change. The content of this review can summarize below. We aim to advance the current insight into the plant pathogenesis and also pave a new way to cope with the upward trends of plant disease by designing the pathobiome-targeted measure